Okay, then let me first start by thanking Liana, Giovanni, and Chris for inviting me to present today's seminar, or webinar, I'm sorry. I really appreciate all your efforts to keep us connected throughout this pandemic. But at the same time, I must confess that having this talk brandmarked as SphingoLeader presentation gives me some difficulties to breathe, given the, I think, excellent lineup of Sphingolipid webinar speakers that came before me, but I will try to give it my best shot. So I believe an important but only slowly emerging concept is that cellular organelles contain information that is encoded in their lipid bilayers. And these so-called lipid codes are critical for specifying organelle identity and function. Now, cracking these lipid codes, I believe, is very challenging. And the first reason for this is that lipids, unlike proteins, are the product of a highly interconnected metabolic network. And that means that that it is very hard to eliminate one particular lipid species without affecting the levels of countless others. And the second reason why this is so hard to do is because lipid codes rely not only on specific lipid species, but also on the collective properties of the bulk lipids in the bilayer. And thirdly, cells use multiple control mechanisms to preserve an optimal lipid composition for each organelle. Now, for instance, the lipid composition of the endoplasmic reticulum, the ER, is characterized by a low Sphingolipid and sterol content, a loose lipid packing, and a neutral cytosolic surface charge. And this is in contrast to the plasma membrane, which has a high Sphingolipid and sterol content, a tight lipid packing, and a highly negative cytosolic surface charge. And these contrasting physical properties, they are reflected in the length and the geometry of the membrane spans of ER and plasma membrane resonant proteins, but they also permit an organelle-specific recruitment of peripheral membrane proteins, such as the small GTPases SAR1 and KRAS, with SAR1 controlling COP2 vesicle formation at the ER, and KRAS controlling cell proliferation at the plasma membrane. And therefore, it's really crucial that cells avoid lipid mixing between the ER and plasma membrane, as this would destroy the lipid codes of these organelles, which I think in turn would likely result in promiscuous interactions, or even worse, a complete breakdown of law and order, as depicted here, with some apologies for the nudity on display. Now, work in my lab is increasingly dedicated to address the functional consequences of and the cellular responses to disease-related imbalances in organellar lipid codes, with a primary focus on the aberrant distributions of sphingolipids. And as I hope to convey in this webinar, our efforts, they have led to some novel insights in, for example, the working mechanism of the tumor suppressor lipid ceramide, on how perturbations in the ER lipid code may cause a rare form of osteoporosis. And last but not least, I would like to revisit a topic that has been introduced already by a previous webinar speaker on the discovery of a novel lysosomal repair pathway in which sphingolipid topology appears to play a decisive role. So let me start by saying that cells, as far as I am concerned, they produce sphingolipids perhaps first and foremost to make their membranes impermeable for solutes. And while sphingolipids are required for cell survival, their production relies on potentially toxic metabolic intermediates, namely on ceramides. And the groundbreaking work by Lina Obeid and Yusuf Hanoun in the 1990s already showed that ceramides are important mediators of stress signaling and apoptosis. So this confronts cells actually with a very nasty dilemma. If they don't make sphingolipids, they die of leaky membranes. And if they do make sphingolipids, they take the risk of killing themselves in the process. So what then are the molecular principles by which ceramides kill cells, and what mechanisms are in place to prevent this from happening, so to make sphingolipid biosynthesis safe? These are some of the questions I would like to go into a bit deeper. Now, as you know, ceramides are synthesized de novo on the cytosolic surface of the ER, and in mammals, the bulk amount of newly synthesized ceramides is transferred to the trans-Golgi by a cytosolic ceramide transfer protein called CERT for conversion into sphingomyelin by a Golgi resident enzyme named sphingomyelin synthase, SMS1. So it appears that newly synthesized ceramides largely bypass vesicular trafficking to reach the Golgi, where they run in a so-called metabolic trap that is set by sphingomyelin synthase 1. And this arrangement has two obvious advantages. First, it helps cells to establish a sharp Sphingolipid concentration gradient along the secretory pathway. And second, it allows cells to readily clear the ER from potentially toxic ceramides as soon as they are made, which seems like a healthy thing to do if you consider that the ER is an organelle that sort of reaches out in all dark corners of the cell. Now, besides the Golgi resonant sphingomyelin synthase, mammalian cells also contain a sphingomyelin synthase-related enzyme, SMSR, which localizes to the ER. And unlike sphingomyelin synthase, this SMSR does not produce sphingomyelin, but instead synthesizes trace amounts of the sphingomyelin analog, ceramide phosphoetanolamine. And already quite some years ago, we found that acute disruption of SMSR catalytic activity causes a rise in ER ceramides and a mislocalization to mitochondria, triggering a mitochondrial pathway of apoptosis. And these findings raised a couple of questions. How does this SMSR control ER ceramides to prevent ceramide-mediated cell death? How do these ER ceramides reach mitochondria upon inactivation of SMSR? And how do ceramides upon their arrival in mitochondria commit cells to death? And in this part of this webinar, I would like to primarily focus on that third question and discuss some of the tools that we have used to dissect the mechanism by which ceramides trigger cell death. So, for example, how do we actually know that the arrival of ER ceramides in mitochondria is a critical step in committing these SMSR-deficient cells to death? Well, the first indication for this was that targeting a bacterial ceramidase to mitochondria rescued the SMSR-deficient cells from undergoing apoptosis. And this result also implied that it was the intact ceramides and not their breakdown products that are responsible for inducing apoptosis. And to confirm that the arrival of ER ceramides in mitochondria is sufficient to commit cells to death, we also studied the consequences of targeting CERT-mediated ceramide transport to mitochondria. And to this end, we substituted the Golgi targeting pH domain of CERT for an automitochondrial anchor, membrane anchor, to create so-called mitocert. And we found that expression of mitocert on its own triggers a mitochondrial pathway of apoptosis, and that this pathway is also strictly dependent on the pro-apoptotic BCL2 protein BAX. Now, indeed, as shown here, expression of mitocert induces mitochondrial translocation of BAX, and in contrast, mitocert aversion that lacks the ceramide binding domain failed to trigger BAX translocation. Moreover, if cells are treated with inhibitors of de novo ceramide synthesis, myriosin, also the mitocert-induced BAX translocation could be blocked. And all of this indicated that a rise in mitochondrial ceramides activates BAX, triggering mitochondrial apoptosis. But an important question then was whether the ceramide-mediated activation of BAX relies on direct or specific interactions between BAX and ceramides, or whether this process also requires other mitochondria resident proteins. And to address this question, we used the photoactivatable and clickable ceramide analog in a fishing expedition for mitochondria resident ceramide binding proteins. Now, photoaffinity labeling experiments on mitochondria consistently revealed a mitochondrial protein of 33 kilodalton that was very efficiently cross-linked to our ceramide probe. And mass spec analysis of the affinity purified cross-linked lipid protein complex showed that this 33 kilodalton protein actually corresponds to the voltage-dependent anion channels VDAC1 and 2. Now, VDACs, they reside in the outer mitochondrial membrane, where they play a crucial role in metabolic crosstalk between the cytosol and mitochondria. But in addition, VDACs also function as mitochondrial translocation platforms for both pro-apoptotic proteins such as BACs, as well as anti-apoptotic proteins such as hexokinases. And to search for a putative ceramide binding site on these VDACs, we started a collaboration with Manel Melo and Sivertjan Marink in Groningen, and they performed so-called coarse-grained molecular dynamics simulations of VDAC channels in model bilayers that mimic the outer mitochondrial membrane and that contained 5% more of C16 ceramide molecules. And in agreement with the affinity labeling, photolabeling data, VDAC1 and 2, but not VDAC3, were found to have a binding site for ceramide in the bilayer center on one side of the beta barrel. And here you see a movie playing of a VDAC1 channel with a single ceramide molecule that you can see is probing the channel's outer wall until it homes in on a binding site that is buried in the membrane's interior. And strikingly, an acidic residue that faces the lipid bilayer seemed to promote a direct contact with the ceramide head group and the channel. Now, it turns out that this particular residue corresponds to a glutamate that is positioned in the middle of the fourth beta strand in VDAC1 and 2. And of note is that the same glutamate had previously been implicated in the binding of hexokinase, which induces channel closure and prevents BAX-mediated apoptosis. And also relevant is that in VDAC3, which does not bind ceramide, this glutamate residue is replaced by a glutamine. And moreover, the simulations show that if you substitute this membrane-facing glutamate for glutamine, then the ceramide occupancy during this particular simulation is strongly reduced, and also the time the ceramide is lingering at that binding site is drastically shortened with at least 1 order of magnitude. Now, to verify that the membrane-facing glutamate residue in VDAC1 and 2 also promotes ceramide binding in reality, we carried out ceramide photolabeling experiments on the wild-type and glutamate substitution mutants of recombinant channel proteins that were reconstituted in proteoliposomes. And as you can see here, in line with the molecular dynamics simulation, substitution of the glutamate for a glutamine abolished photolabeling of both VDAC1 as well as VDAC2. And these results sort of confirm that the membrane-facing glutamate in VDAC1 and 2 is a critical determinant of ceramide binding. Now, photolabeling of these VDAC channels with the ceramide probe was also progressively inhibited when carried out in the presence of excess of native C16 ceramide molecules, while in contrast, addition of an excess of C16 ceramide had no impact on photolabeling of VDACs with a photoactivatable cholesterol probe. And I forgot to mention that also in simulations, the VDAC contained multiple hotspots of cholesterol binding, but they mostly fell outside the ceramide binding site. So this confirmed that the membrane-facing glutamate is part of an authentic ceramide binding site. Now, previous work by the Abramson lab showed that the membrane-facing glutamate in VDACs and VDAC1 has a pKa of around 7.4, which is close to the pH of the cytosol. And this finding implies that under normal physiological conditions, this glutamate, even though buried in the membrane bilayer, is in its fully charged, deprotonated state at least a significant amount of time. Protonation of that membrane-facing glutamate was found to actually enhance the formation of VDAC1 dimers, presumably because some hydrogen bonding occurs between the glutamate and a serine residue at the dimer interface, which then stabilizes formation of a dimer. And in simulations, we found that protonation of the membrane-facing glutamate in VDAC1 and 2 also blocked ceramide binding, And also in line with this a lowering of the pH from 7 to 5 also significantly reduced the photolabeling of the recombinant VDAC channels with the ceramide probe. So this all indicated that ceramide binding to VDACs relies on the protonation state of the membrane-facing glutamate and occurs only when this residue is in its deprotonated, fully charged state. So what is the biological implication of these biological studies, biochemical studies, sorry. So what we propose is that VDACs serve as ceramide-dependent modulators of apoptosis. So as I said before, these VDACs interact with both anti-apoptotic proteins, such as hexokinases, as well as with pro-apoptotic proteins, such as Bachs. Hexokinase binding to these VDACs is believed to antagonize cell death by inhibiting Bach's-induced release of cytochrome C. And in addition, the overexpression of these hexokinases and their association with VDACs, they are typical features of hyperglycolytic cancer cells. And when bound to VDACs, these hexokinases are able to couple oxidative phosphorylation to glycolysis by capturing ATP released from mitochondria to phosphorylate glucose, which is the rate-limiting step in the glycolytic cascade. Now, in the clinic, there's actually quite some interest in drugs that dissociate hexokinases from VDACs, as such drugs would have a dual anti-neoplastic effect. First, they would facilitate induction of apoptosis, and second, they may revert the hyperglycolytic state that allows tumor cells to proliferate in areas of the body where oxygen is in short supply. Now, our finding that ceramides bind VDACs at a site that overlaps with the binding site for hexokinases implies that ceramides may exert their pro-apoptotic activity, at least by influencing the interaction between VDACs and hexokinases. And this is a model that we are currently putting to the test. Our first approach to this extent was that we asked whether VDACs play a role in ceramide-mediated cell death. And by monitoring caspase-mediated cleavage of PARP1, we found that loss of VDAC2 rendered cells partially resistant to mitoCERT-induced apoptosis, especially when cells also lacked VDAC1. So suggesting that VDAC2 plays a role in this mitoCERT-induced pathway. And importantly, reintroducing the VDAC2 in a VDAC1 double knockout could restore the ability of mitoCERT to trigger apoptosis as judged by monitoring PARP1 cleavage and proteolytic activation of caspase-3 here. In contrast, reintroducing VDAC1 had little effect on this pathway, and this qualified VDAC2 as the major effector protein in the ceramide-mediated cell death pathway. So importantly, substitution of this membrane-exposed glutamate in VDAC2 also disrupted the ability of that protein to restore mitoCERT-mediated apoptosis. So this indicates that ceramide requires a ceramide-binding competent form of VDAC2 to trigger cell death. Now, consistent with this, with our model, Dina Hassan in our group showed that the membrane-buried glutamate in VDAC1 and 2 is essential for mitochondrial recruitment of hexokinase 1. And this is shown in these experiments where she looked at the GFP-tagged hexokinase and that they fall off from mitochondria when either both VDACs are removed or when instead of a wild-type VDAC, a VDAC point mutant that lacks the glutamate is reintroduced. And this is the Pearson coefficient data on those experiments. And moreover, Dina also found that the enzyme's short hydrophobic N-terminal alpha helix likely directly participates in VDAC binding, presumably by inserting itself into the hydrophobic core of the bilayer with potentially a methionine at the N-terminus in direct contact with the glutamate. So we speculate that binding of ceramides to the negatively charged glutamate in VDACs may prevent hexokinases from binding and thereby lowering the threshold for VDAC oligomerization at neutral pH, which would occur at that same interface. And such ceramide-triggered oligomerization of VDACs may then also trigger apoptosis, by, on the one hand, blocking binding of anti-apoptotic hexokinase, and on the other, by creating a suitable platform for mitochondrial translocation of BACs. So, to challenge this model, Marcus Schneider in our lab is pursuing a bottom-up approach in which he uses reflectometric interference and TIRF spectroscopy to measure the interaction kinetics between VDAC channels embedded in polymer-supported bilayers and their fluorescent-labeled protein binding partners, such as hexokinase 1. And here's an example of a typical binding experiment whereby the RIF intensity signal, as shown here, allows you to monitor how the VDAC-containing proteoliposomes, when injected into the system, form a stable, defect-free planar bilayer, polymer-supported bilayer, and then subsequent injection of the fluorescent-labeled hexokinase causes then an additional increase in the RIF signal that is not observed when one injects a control protein. Now, binding of hexokinase to these VDAC containing bilayers is also detected in the TIRF spectroscopy channel with a much higher signal-to-noise ratio in comparison to the RIF channel. And pseudo first-order association and dissociation rates can eventually be extracted by these curves and then provide a quantitative assessment of the association and dissociation of the hexokinases for these VDAC-containing membranes. Now, experiments to investigate the impact of ceramides on these binding kinetics are currently in progress. And this brings me to some first summary. So what I showed you was that ceramides seem to be able to exert apoptotic activity directly in mitochondria, that VDAC2 seems to be a critical effector of ceramide-mediated cell death, and thirdly, that ceramide binding on this VDAC overlaps with the binding site of hexokinases, and that this points at a potential mechanism by which ceramides exert their tumor suppressor activity, both with respect to triggering cell death and preventing a hyperglycolytic state of cancer cells. Now, this brings me to the second topic of this webinar. I already mentioned that bulk production of sphingomyelin, which is the key sphingolipid in mammalian cells, is mediated by this Golgi-resident enzyme sphingomyelin synthase 2, but in fact, mammalian cells have a second sphingomyelin synthase isoform, SMS2, which actually dwells both in the trans-Golgi and at the plasma membrane of these cells. Now, the sphingomyelin produced by these enzymes at these locations provide a sink for cholesterol synthesized in the ER, and so this arrangement contributes to the formation of a sphingolipid sterol gradient along the secretory pathway, which is important for maintaining ER and plasma membrane-specific lipid codes. Now, in collaboration with a group of Utti Makiti at the University of Helsinki, we found that actually SMS2 is highly expressed in bone, and we also identified heterozygous mutations in the SMS2 gene as the underlying cause of a rare form of osteoporosis, which is characterized by sclerotic lesions in the skull of the patients that are affected by the disease. And these mutations turned out to block export of a functional sphingomyelin synthase enzyme from the ER. And based on these findings, we postulate that the cause of osteoporosis in these patients with SMS2 mutations is not a reduced cellular capacity to produce sphingomyelin, but rather a disrupted sphingomyelin sterol gradient due to an aberrant bulk production of sphingomyelin in the ER of the osteogenic cells. Now, to challenge this hypothesis, Jan Parolek and Tolulupe Sukoya created a toolbox of disease-relevant cell lines in which wild-type or pathogenic SMS2 variants were put under control of a doxycycline-inducible promoter. And they found that doxycycline induced the expression of pathogenic SMS2 variants, that this fully restored sphingomyelin levels in cells that lack endogenous sphingomyelin synthase 1 and 2 enzymes. And they also found a remarkable 3-4-fold rise in sphingomyelin species containing a dihydroceramide backbone, which likely reflects a competition between SMS2 resident enzymes of sphingomyelin synthase enzymes and the ceramide desaturase for dihydroceramide substrates. So all in all, this type of experiments confirmed that in cells from patients with these heterozygous SMS2 mutations, bulk production of sphingomyelin is mislocalized to the ER. To investigate whether expression of pathogenic SMS2 variants also alters the subcellular distribution of sphingomyelin, Tolu next exploited an engineered variant of the sphingomyelin binding toxin equinatoxin, which was developed in a group of Christopher Bird to monitor intracellular sphingomyelin trafficking. And this non-toxic equinatoxin variant readily stains the cell surface when added externally, but it does not stain any membrane-bound organelle when expressed as a cytosolic protein. And in line with this strict sphingomyelin asymmetry, indeed, sphingomyelin produced by the wild-type SMS enzymes accumulates in the exoplasmic leaflet of late secretory and endosomal organelles, but it does not show up in the cytoplasmic leaflet as shown here in the cells that express the cytosolic version of the sphingomyelin reporter. But in contrast, in cells that express the pathogenic SMS2 variant, the cytosolic sphingomyelin reporter is readily recruited to intracellular vesicular structures, the identity of which we have not been able to establish. But these sphingomyelin positive structures also largely stain positive for the cholesterol reporter D4H, which normally stains primarily the inner leaflet of the plasma membrane. And from this, we conclude that expression of pathogenic SMS2 variants actually causes a major assault on the lipid code of early secretory organelles, which includes a break in sphingomyelin asymmetry and a major redistribution of cholesterol. Now, according to the model that I presented at the beginning of my talk, such an assault on the lipid code of secretory organelles should have also a devastating effect on probably every cell in the body of a patient that carries such pathogenic SMS2 mutation. But this apparently is not the case. So why do these patients primarily suffer from a bone disorder? Well, as I said, SMS2 is highly expressed in bone, and so it's fair to anticipate that its mislocalization would be particularly detrimental for osteogenic cells. And these cells happen to produce the largest amounts of type 1 collagen anywhere in the body. And this type 1 collagen is a bone critical protein that comprises about 80% of the total protein found in bone. And collagen secretion is actually a highly demanding process as it relies on a correct assembly of pro-collagen trimers in the ER and a subsequent incorporation in enlarged COP2 transport carriers. And formation of these carriers requires proteins like SAR1, the GTPase, as well as specialized transport machinery that includes Tango1, a protein that helps coordinate COP2 bud expansion to accommodate the large collagen trimers. Mutations on COP2 subunits, which otherwise do not interfere much with protein secretion, have been shown to disrupt collagen secretion and cause major ossification defects in the skull of patients that carry such mutations. And we therefore postulate that secretion of collagen may be particularly vulnerable to alterations in the ER lipid code invoked by pathogenic SMS2 variants. And this is why Tolu is currently setting out to investigate the maturation and secretion of collagen in osteogenic cells that express pathogenic SMS2 variants and analyze the bone forming capacity of these cells in 3D hydrogel bone models with our collaboration partners. And then this brings me to the third and final part of this webinar, in which I would like to highlight a previously unrecognized but a very important functional aspect of sphingomyelin asymmetry. Consistent with a recent study by the group of Felix Rando, Patrick Niekamp, which was a previous webinar speaker, found that not only pathogenic SMS2 variants, but also the invasion of macrophages by pathogenic microbacteria can trigger a break in sphingomyelin asymmetry, namely at the sites where the microbacteria escape from the phagosome and enter the cytosol. no cytosolic sphingomyelin exposure actually can be found when the macrophages are infected with a vacuolar translocation defect or bacterial mutant strain, so supporting this assessment. Now, when Patrick treated cells with the lysosome damaging drug LLOME, what he found is that the cytosolic sphingomyelin report is also readily accumulated in numerous puncta that also became gradually positive for galactin-3, which is a lectin binding the luminal glycans of lysosomes when this integrity of lysosomes is destroyed. But importantly, the sphingomyelin reporter was recruited to damage lysosomes prior to galactin-3 recruitment with a time difference of about 5 to 6 minutes. And collectively, his data and those reported by Felix Rando's group indicate that a break in sphingomyelin asymmetry is an early marker of lysosomal damage that precedes the catastrophic breakdown of lysosome integrity when the luminal glycans get exposed and recognized by the cytosolic galactins. So it seems that lysosome damaging drugs and the translocation performed by pathogenic mycobacteria initially cause only relatively minor perturbations in the lysosomal bilayer that somehow facilitate the transfer of sphingomyelin from the luminal to the cytosolic leaflet before progressing in a complete collapse of the membrane barrier and the cytosolic exposure of the glycans. And the question is, what is the molecular principle behind this sphingomyelin transfer? But before I go into this, let me also show this particular experiment where it turns out that the cytosolic sphingomyelin reporter is also readily mobilized to the plasma membrane when the plasma membrane is wounded by a pulse laser. And this indicates that, and this actually occurs almost instantly after laser light is induced on the cell. And this indicates that a break in sphingomyelin asymmetry is associated, in fact, with a wide range of injuries and multiple organelles. And the question again is, what are the molecular principles that drive this damage-induced sphingomyelin translocation? Now, as you have heard, an influx of calcium has been widely recognized as a key trigger in the detection and repair of membrane injuries. So we wondered whether calcium also played a role in the damage-induced translocation of sphingomyelin. And the first indication that this indeed may be the case was a finding that cells pre-treated with the lysosomal calcium chelator BAPTA show a greatly reduced reduction in the number of equinotoxin positive puncta that cells show in response to the lysosome damages drug LLOME when compared to controls. Moreover, when these cells were treated with the calcium ionophore ionomycin, the cytosolic sphingomyelin reporter was also readily accumulated in countless puncta. And importantly, the formation of these puncta was strictly dependent on the presence of calcium in the culture medium. When calcium is depleted, none of these puncta would form. Now, based on these findings, we conclude that the damage-induced sphingomyelin translocation is a calcium-dependent process and presumably involves a calcium-activated lipid scramblase. So the idea would then be that any type of damage that causes a minor injury of the lysosomal or plasma membrane would trigger an influx of calcium into the cytosol, and a rise in cytosolic calcium would then activate the scramblases near the site of membrane damage, and this would then lead to a local exposure of sphingomyelin on the cytosolic surface of the damaged organelle, and then mobilization of the sphingomyelin reporter. Now, previous studies by the group of Phyllis Hansen and Harald Stenmark have shown that the SCOT machinery, notably SCOT3 subunits, are recruited to damaged lysosomes and participate in their repair. As SCOT recruitment has been found to be dependent on calcium, we wondered whether cytosolic sphingomyelin exposure may be part of the mechanism by which SCOT is mobilized to injured lysosomes. And as shown here, Patrick found that mobilization of the sphingomyelin reporter to LLOME damaged lysosomes indeed precedes the recruitment of the SCOT3 subunit CHIMP4B, with a time gap of about 30 to 60 seconds. And this raised the possibility that indeed cytosolic sphingomyelin may actually serve as a binding site for escort components. However, to investigate whether sphingomyelin, before I come to that, so to investigate whether sphingomyelin plays a role in the mechanism by which cells detect and repair damaged lysosomes, Patrick actually next monitored the recovery of lysotracker in lysosomes of both control or sphingomyelin synthase knockout cells, hence sphingomyelin-deficient cells, that were transiently exposed to the lysosomotropic drug GPN. And as shown in this graph, lysotracker fluorescence is readily lost after brief exposure to GPN, but then returns after the GPN is washed away. And during this process, the proton gradient required for trapping lysotracker inside the lumen is restored due to the repair of membrane damage. So this is also can be followed here in this particular graph, which showed that, this part is not shown, but that the wild-type and sphingomyelin synthase knockout cells acquire lysotracker to the same extent, but they also lose lysotracker fluorescence with the similar kinetics after adding GPN, which all of this indicated that in principle, their lysosome function is comparable. However, the recovery of lysotracker after removing GPN was significantly delayed in the sphingomyelin synthase knockout cell line in comparison to the wild type. And moreover, far less lysotracker positive structures were recovered in the mutant sphingomyelin synthase mutant cell line than in the wild type. And so in addition, if we take the sphingomyelin synthase knockout cells and then transduce them with a sphingomyelin synthase 1 construct under control of doxycycline and a inducible promoter, the addition of doxycycline was able to restore the capacity of the double knockout to regain lysotracker fluorescence, essentially to the same level as in wild-type cells after GPN is washed away. So these results are clearly pointed at a critical role of sphingomyelin in the mechanism by which cells repair damaged lysosomes. So as cytosolic sphingomyelin exposure proceeds to recruitment of SCOR3 proteins to damage lysosomes, Patrick investigated whether sphingomyelin is required for mobilizing this central SCOR3 component. However, this CHIMP4B recruitment to the lysosomes in LLOM treated sphingomyelin synthase knockout cells occurred with exactly the same kinetics as the wild-type cells, except that we found that the sphingomyelin synthase knockout cells displayed a bit more of a prolonged CHIMP4 retention on the damaged lysosomes. So this suggested that cells are equipped with a sphingomyelin-dependent lysosomal repair pathway that may operate in parallel with the escort pathway. Now, to investigate whether sphingomyelin per se or sphingomyelin exposed on the cytosolic surface of damaged lysosomes is critical for their repair, we then generated a construct in which a bacterial sphingomyelinase was fused to the cytosolic tail of the lysosomal associated membrane protein LAMP1. And as shown in these two movies here, expression of the enzymatically active fusion construct, but not its enzyme dead variant, could efficiently suppress the mobilization of equinotoxin to damage lysosomes in LLOM-treated cells, indicating that the active fusion protein catalyzes a very rapid and efficient turnover of sphingomyelin on the cytosolic surface of damaged lysosomes. And to address whether metabolic turnover of the cytosolic sphingomyelin reduces or promotes the repair of damaged lysosomes, we next analyzed the effect of the active or enzyme-dead sphingomyelinase fusion construct on the ability of the cells to regain lysotracker fluorescence after transient exposure to GPN. And what we found is that expression of the active construct, if anything, promotes the recovery of lysotracker fluorescence. And this effect became even much more pronounced when the experiments were carried out on cells that were depleted of two proteins called ALEX and TSG101, which are two proteins that are essential for escort-3 recruitment to damage lysosomes. So this implied that the exposure and subsequent turnover of sphingomyelin on the cytosolic surface of damaged lysosomes stimulates their repair, even when escort-3 recruitment is blocked. And given that ectopic expression of bacterial sphingomyelinase promotes lysosomal repair, we anticipated that inhibition of endogenous sphingomyelinase that act on cytosolic sphingomyelin pools would reduce lysosomal repair, and this is exactly what we observed. So addition of the generic neutral sphingomyelinase inhibitor GW4869 reduced the recovery of lysotracker fluorescence after transient exposure of GPN. And this inhibitory effect of GW4869 became even much more pronounced when the experiments were carried out on cells that were depleted for ALEX and TSG1. And in line with the critical role of neutral sphingomyelinase in lysosomal repair, we also found that the GW4869 significantly reduced the viability of cells in the presence of these lysosome damaging drugs, LLOME. And finally, this work raised the question which member of the neutral sphingomyelinase enzyme family may actually participate in lysosomal repair. And as shown here, the siRNA-mediated depletion of the neutral sphingomyelinase number 1 had no impact on lysosomal repair. However, depletion of the neutral sphingomyelinase number 2 caused a small but very significant reduction in the lysosomal repair and also rendered cells more sensitive to the lysosome damaging drugs. Now, based on all these findings, we conclude, and now I come to the final concept and outlook slide, that cells appear to be equipped with a Sphingolipid-driven lysosomal repair pathway that operates largely independently of the escort pathway. And here's how we think this pathway works. So we anticipate that any type of damage that disrupts the integrity of the lysosomal membrane causes a leak of calcium from the lumen into the cytosol. And a local rise in cytosolic calcium levels would activate a calcium-dependent scramblase near the site of the membrane damage, thus leading to a local exposure of sphingomyelin on the cytosolic surface of that damaged organelle. This pool of cytosolic sphingomyelin is then rapidly turned over by a neutral sphingomyelinase, presumably neutral sphingomyelinase 2. And this sphingomyelinase cleaves the bulky phosphocholine head group of sphingomyelin to generate ceramide, a lipid with a much smaller and less hydrated head group. Because these ceramides occupy a smaller membrane area than sphingomyelin and lack affinity for sterols, the formation by sphingomyelin turnover also readily triggers their self-aggregation, causing a local condensation of the cytosolic leaflet. And this in turn would drive an inward budding of the bilayer that is like the membrane deformation catalyzed by the escort machinery, which drives formation of interluminal vesicles, by which membrane lesions can be removed from the lysosomal limiting membrane. Now, this ceramide-driven membrane invagination has directly been demonstrated in giant liposomes, and also previous work by the group of Mika Simons actually showed that neutral sphingomyelinase play a role in the formation of interluminal vesicles that occur in multivesicular endosomes. Now, while the identity of the calcium-activated scramblase involved in this lysosomal repair pathway remains to be established, we anticipate that a very similar pathway is responsible for trapping damaged portions of the plasma membrane and shredding these as extracellular vesicles, but this requires to be shown. So what we are postulating is that the strict sphingomyelin asymmetry in late secretory and endosomal organelles actually reflect a vital role of sphingomyelin and neutral sphingomyelinase in safeguarding the functional integrity of these organelles. Okay, and with this I come to an end of my webinar. I would like to acknowledge all past and current members of my lab, the EU and DFG for funding our projects, and our international collaborators with whom we started a range of adventurous projects, such as with the Trauner lab regarding the application of photoswitchable sphingolipids, the MELO lab with respect to molecular dynamic simulations, the Markiti lab regarding sphingolipid metabolic enzymes and bone disease, and the Tafessa and Barish labs regarding the role of sphingolipids in mycobacterial infection. But I also like to acknowledge Christopher Bird and Christopher Clark for their support in characterizing this novel sphingolipid-driven lysosomal repair pathway. Now, of course, webinars are great to stay connected during a pandemic and may even help us to reach our climate goals faster. However, I believe they are a poor substitute for throwing in an after party in one's hotel room to hang out just a little bit longer with the people in your field that you respect and cherish. And because we cannot have enough scientists that inspire countless others and will stay forever young, I would like to dedicate this webinar to Lina Obeid in the hope that she would approve that. Thank you very much for hanging out with me.